Hi, my name is Dylan Tomasoff and I am a biology major at CSU Fresno. Do you know what the most common physical disability is in children? Would you have guessed that it is cerebral palsy and the prevalence of the disease has remained stable at 2 to 3 per 1,000 live births for the last 4 decades? Yet we are still lacking the knowledge of what the mechanism that drives the development of this non-progressive disease. Due to the lack of a known mechanism, I will describe how the disease presents itself and the leading hypotheses of both genetic and the possible correlated conditions from in utero to neonatal that seem to cause cerebral palsy. The clinical descriptive term cerebral palsy is a term applied to a heterogeneous group of neurodevelopmental disorders that present with various symptoms affecting motor function and a wide range of other medical disorders. Cerebral palsy has 4 major components that include the type and the severity of motor abnormalities, anatomical distributions, associated impairments, and the time in which the presumed causal event occurred: prenatal, perinatal, or postnatal. There are 2 different neuromuscular tones, hypotonia and hypertonia. The next characteristic is the predominant type of motor impairment, which are spastic, atastic, dyskinetic, and or mixed. The involvement of each limb or trunk differentiates unilateral or bilateral anatomical distributions, which are monoplegia, diplegia, triplegia, hemiplegia, and tetraplegia. Besides the various neuromuscular issues, the disease is commonly found to have comorbidities. The comorbidities are intellectual disabilities in 30 to 65%, seizure disorders in 30 to 50%, speech and language deficiencies in 40%, visual impairments in 40%, and hearing issues in 5 to 15%, and 9% of patients have autism spectrum disorder. The leading hypothesis for cerebral palsy is complications during labor and delivery, and the other hypotheses occur in utero. There is a growing body of evidence that is pointing at multiple genetic factors that results are still inconclusive with. With the use of population-based genetic studies, there have been several candidate genes identified, with some of them being associated with processes of inflammation, coagulation, and blood flow. This study showed that genetics seems to play 24% for preterm and 48% for term. There are 3 types of cerebral palsy that show a strong inheritance pattern through an autosomal recessive means. The 3 types of cerebral palsy are atastic cerebral palsy, symmetrical spastic cerebral palsy, and tetraplegic cerebral palsy, even though these forms only make up a small percent of cerebral palsy cases. Within the same peer-reviewed article, it is discussed a family that had a repeating congenital cerebral palsy with a candidate gene that is only expressed through the father, but no mention of if this is a Y-linked inheritance pattern. Another leading hypothesis is epigenetics, to make possible connections on what is causing things such as the repressive effects of microglia, astrocytes, overactivation, though this is not the only viable hypothesis, as others tend to focus on prenatal mechanisms. There are 3 main categories that I will discuss. They are the predisposing pathological processes, peripartum events, and neonatal complications. The author of this article proposed a double hit model that suggests that an intrauterine condition and intrapartum and postnatal insult can lead to the development of cerebral palsy. Risk factors of cerebral palsy include congenital malformations, fetal growth restriction, multiple gestations, infection during the fetal or neonatal period, birth asphyxia, preterm delivery, untreated maternal hyperthyroidism, perinatal stroke, and thrombophilia. The intrauterine process that is linked with fetal growth restriction also led to neuronal damage and resulting cerebral palsy, yet the association between preeclampsia and the development of cerebral palsy is under constant debate. Moving on to prenatal stroke, which over 10 years of a study showed that 39% of 100 neonates diagnosed with neonatal arterial ischemic stroke went on to develop cerebral palsy. Another possible link is with placental lesions. There are three different types of placental lesions. They are evidence of infection, inflammation, maternal underperfusion, and fetal vascular thrombo-occlusive disease that contributed to the association between these specific placental lesions and the development of cerebral palsy. I will now move on to encephalopathy and prematurity, as there is a quantitative measure of gray and white matter lesions that correlate to unilateral cerebral palsy. PVL is an injury to the white matter and is a strong prognostic factor in developing cerebral palsy. Reactive gilios excitotoxicity, microglia activation, the release of free radicals, and subsequent hypomyelination are associated with PVL and are divided into the category of diffuse and cystic, whereas injury to the gray matter that causes abnormal somatosensory inputs may be a causal factor of motor impairments due to neuronal and axonal degeneration, including the somatosensory pathway. Though both fetal growth restriction and birth asphyxia are greater risk factors to developing cerebral palsy, they lack evidence of causality, unlike intra-amniotic infection and inflammation between PVL and interuterine processes, while neonates with PVL had an increased immunoreactivity for TNF-alpha in the neocortex, hippocampus, basal ganglia, and thalamus. Yet another possible cause is mothers who had fevers during labor, which gives a 9-fold increase in the risk of developing cerebral palsy. Encephalopathy of prematurity is the leading risk for developing cerebral palsy, with 60% of those with EOP going on to develop cerebral palsy. EOP is a complex mix of both destructive and developmental disturbances involving both gray and WMI as a result of interuterine hypoxia, ischemia, and or systematic infection/inflammation.